It's going to be kind of fun because we're going to be going from 4,000 meters above sea level to 4,000 meters below sea level. So we'll get to see some of the differences and how humans might do in that environment. So I'm going to talk about this deep sea habitat, and I'm going to talk a little bit about some of the different inhabitants of that environment that may not be familiar with you, and then some of the adaptations that they have, that they share. So we're all familiar, I'm representing the Earth here as this little brown line. The ocean has, it covers the globe, and we define the deep sea basically as most of the ocean below about 200 meters, so about below the penetration of light. And there's also the ocean bottom, which a lot of deep sea biology is related to just the ocean bottom, but I'm actually interested in this environment here. Just to give you a sense of scale relative to the Empire State Building, for example, the deep sea, just this part of the deep sea is about 10 Empire State Buildings deep, about twice as deep as the Grand Canyon, and that's the average depth of the ocean throughout the world. This isn't the deepest part of it. The deepest part is about 2.5 times that. So just the average depth of the ocean, if you go out into the middle of the Pacific or Atlantic, is about 4,000 meters. So what are sort of the physical characteristics of this habitat? Well, one, of course, is the temperature. We all know that it's cold down there. The average is between 2 to 4 degrees Celsius, so it's quite chilly. And we also know that light in the ocean is initially filtered to these sort of blue wavelengths of light, but then as you go down, of course that just dims out, and so most of the ocean below about 250 meters is in darkness, and the only light that you get is biologically generated, and I'll talk more about that. And then the other obvious one is the pressure, and so we've just seen this sort of high altitude, the inverse of this, but if you go down into the deep sea, it's an atmosphere for every 10 meters, and so there's a very, very large pressure at this depth of 4,000 meters. So when I talk about these animals, people are often wondering, how they survive in these pressures. Well, if you think about sort of this sort of cubic inch, the pressure impinging on that is again, it's about 6,000 pounds per square inch. But if you take a water balloon or a Ziploc bag and fill it up with water and you take it down into the deep sea, it doesn't get crushed by the pressure of water molecules outside because there's basically water inside as well. And this is the same thing that the organisms are doing. They're mostly like us, they're mostly water, but unlike us, they don't have these gas spaces that sort of limit our ability to operate at those pressures. So the other thing that is curious about these organisms is when we study them, we go down and we get them and we bring them back up to the surface. So we're basically releasing that pressure. And you expect, like with fish, they have a swim bladder, so their eyes bug out, and they have these problems with that release of pressure. But actually, the deep sea organisms, they're fairly resistant to this change in pressure, and they're not doing it by resisting, but they're just doing it by accommodating, right? They're doing it by having the same pressure inside as is outside. And this is actually the same way with our ROVs, our vehicles that we use, and I'll show you in a sec. We have sort of two categories of trying to deal with the pressure for the vehicle. One is to make this really strong container that can resist the pressure and have air inside of it, but the other is just to fill cables with oil and just have it at the pressure that the environment is at. So you have liquid inside, liquid outside, and it can handle the pressure without any problems. So I said that there aren't gas spaces, but actually there are some interesting things that we're looking at. One of them is this creature that lives at about 2,500 meters, and I'll talk more in detail about this group. It's called siphonophores, but essentially for the moment you can think of this just as a jellyfish, but you can see it has a little gas float at the top. And so it's pretty amazing to us that this can evolve a gas at 200 atmospheres of pressure. You can see some of the little bubbles coming out here when we bring this thing up to the surface. So one of the things we're interested in is figuring out, what is the exact composition of that gas and how you could evolve it at such high pressures. So we have this habitat. It's cold, it's dark, it's high pressure, and it seems sort of far away and foreign to us. One thing you hear a lot, people say, well, the ocean is 70% of the planet, the ocean is 2/3 of the planet. Those are all lies. Don't believe any of that, because we don't operate in a two-dimensional space, right? We operate in this three-dimensional volume. So like I showed you, we have 4,000 meters of space that covers 70% of the planet. So when you look at it in the context of volume, the ocean is actually much, much larger, and actually most of that is the deep sea. So here's the 200-meter portion, and then this 4,000-meter portion. So there's this huge portion of the globe, almost 99% of the space on the Earth is in the ocean, and of that 93% is deep sea. So we have this habitat that is very, it's extreme, but it's sort of context dependent. It's not really exotic in the sense that that is most of the space on Earth. So the question becomes, biologically, what's going on in there? What are the organisms that live in that environment, and what are they doing? So the ways that we study this, the traditional way to do it is to take a net and drag it through the water and just see what comes up in the mesh in the end. And a friend of mine likened this to sort of flying a helicopter with a grappling hook over a city and just seeing what comes up on your grappling hook. If you're lucky, you would get some hats and umbrellas and things, and if you're unlucky, you might get something worse. But it kind of gives you a skewed picture of that environment because of your ability to observe it. And so using these nets, we come up with sort of the typical idea people think of living in the water column, sort of shrimp-like things, and these are called copepods. These are sort of the insects of the ocean in a way. They're the kind of arthropods, the bugs that live down there. Your net may also come up with some weird parts that you may either just sort out and throw away, or not really be able to identify. And using these techniques, you're never going to know that sort of this Humpty Dumpty is put back together into this organism. Those parts fit back together into this whole organism. So how do we get around that? There's a few ways that we can do that. One is by actually going down in a manned submersible and collecting organisms using these samplers. And at my institute, we use a lot of remotely operated vehicles. And so this is one of the subs where we're on the boat. There's a tether here that sends power and gets communication both from and to the vehicle with high-definition video cameras and a bunch of other video cameras. So we can just be sitting in the ship controlling this almost like a video game, or literally with a joystick, controlling the vehicle and the cameras, and we can collect these fragile organisms that live in the deep sea fairly intact. So here's an example of another jelly, and here's the sampler, and what's actually happening, this sort of SUV-sized vehicle is being driven over, and so now we're driving down, and then we can close the lid, and so you can catch the jelly. So it's not being suctioned into that tube, but we're just catching that whole little volume of water. And that species in particular, those tentacles, it shoots them off, it lets go of them whenever it's disturbed. So all the descriptions that you'll find in the literature are, this jelly that has tentacle stubs around it, but not the whole thing. So we can collect whole organisms like that. We can also do almost underwater surgery in a way. This is a comb jelly, which I'll tell you more about in detail, but it's about the size of a football, and it's one of the groups of organisms that I particularly work on. And so I wanted a DNA sample from this, and so we put it into the sampler, and I sort of saw how big it is. All this white stuff is the eggs, and so this is a sort of a ripe, version, and I felt really bad because I just needed, literally a molecule from this. So I thought, well, let's just let it go, and then just grab the tip of the tentacle with the lid of the sampler, and just slice it off. So that was all fine in theory. But as we drove away, we found out how strong and stretchy those tentacles can be. So we sort of flew this ctenophore kite around underwater for a little while. And then finally the tentacle broke off, and we were able to get the DNA sample. And actually it's in the tree that I'll show you a little bit later. So these are our tools basically to explore these relatively unexplored inhabitants of that large deep sea habitat. So what do we find down there? One thing, when you think about jellies, you think about these gelatinous plankton. Probably these large scyphozoans as a class of cnidarians come to mind. There are scyphozoans down there, not really of this type, but they're actually relatively small proportion of the kind of inhabitants that we see. We see a lot more of these hydromedusae. These are small transparent jellies. And this is an example of one that a friend of mine described called Chrysota millsii. And you can see, again, these super fine tentacles. And this one feeds on very small things in the water column. So it sends out this big mesh of tentacles. And this was only just described about 4 years ago because you just can't get those fragile organisms in any kind of condition. So within the Hydromedusae, though, there's actually this whole other group of organisms that we see it all the time. It's one of the more abundant kinds of organisms down there. But even in marine biology textbooks, they may get, a paragraph or something because they're just really unfamiliar. And these are the siphonophores. So siphon means tube and phore means bearing. So these are sort of the tube creatures. The one that you probably have heard of is the Portuguese man-o-war, but this is a very unusual form. Most of the ones that we find in the deep sea are more like this, or even in the shallow ocean. They have these swimming bells, these sort of little rocket propulsion modules, and then they have, connected by this tube, they have all their stomachs and their tentacles in a separate part. So this is one of the adaptations that we see a lot of in the ocean, is that they're not really constrained to a small space. They don't have to deal with any kind of boundaries or borders, and so they can grow to be extremely long. And actually the longest organism on Earth, at least in terms of animal, is thought to be this siphonophore right here, Praia. It has 2 of those little swimming modules, and then all these yellow things are stomachs and their tentacles and their reproductive structures. And so it's just straight out in a tube behind it, and that just these 2 parts at the front are what move it in and pull it along through the water. These parts by themselves can't feed. The stomachs that are down the way are sort of the feeding units, and they send the nutrients back up through this tube. So it has specialized zooids along the length of it that allow it to function as a whole organism. And in this case, you can see the tentacles hanging down. These little yellow specks are the stinging cells on these tentacles, and they're about 2 meters long. So you have something that would be maybe 40 meters long with 2 meter long tentacles hanging down, one organism. And this is continuously feeding with krill, pulling krill up into the individual stomachs. So I guess there's no independent control of the lights. So one organism can basically have a very large predatory impact on the environment. And again, these are pretty unfamiliar to probably even most marine biologists, but they're really, really abundant down there, and we just can't get them using most of the normal sampling techniques. They were actually popular. Darwin and Haeckel and a lot of the 19th century biologists were completely familiar with siphonophores, and here's one of Haeckel's plates showing a species that we can still get today. So this is one that I got in Mexico that they did a good job then of trying to appreciate these organisms in their entirety and how they function as an animal, and not just the random bits that you might get back in a net tow. So there are a lot of species of these things. This is the descriptions through the decades, and actually with the ROVs and with our sampling that we have now, these are mostly undescribed. A couple of these have been described since I made this slide, but there's a whole bunch of diversity out there, basically, that is yet to be kind of appreciated and explored. Here's some new. I probably shouldn't call them new species. They've been around for millions of years. I should just call them undescribed species. That's all right. I don't mind this yellow light. It's cool. So these are some of the ones that have some interesting properties. This one has, we call it the blue scythe because it's got these blue iridescent speckles inside of it. This is just from the light that I used to take the photograph. This one has fluorescence, and I don't know if I'm going to have time to talk about fluorescence, but basically a lot of the organisms use fluorescence and bioluminescence. And so this one we named after its property of fluorescence. Here's another one where the sort of tail of it, or the feeding part, is really contracted up and protected by these clear things. The white spots on those are stinging cell, batteries of stinging cells, so it's kind of got this little protective sheath around the tail of it. And this is a deep sea one that's super fragile and small, and it was named after my lab tech, Lynn. So it's kind of a nice jelly for a nice lab tech. One of them that was kind of interesting that the paper is just coming out has, so these yellow things are the side branches on the tentacles, and you can see here an example of one coiled up, and within each of these coils are thousands of those stinging cells that give cnidarians their name. But this one has this weird leafy structure on it that also has fluorescence, and so we think this is essentially a little lure that they've used to increase the amount of, in this case, shrimp that come by to check out their stinging cells with these strange shaped lures. But we're trying to figure out what it is that shrimp like about that shape. I haven't figured that out. So those are the siphonophores, these tube-bearing organisms. They're essentially like a hydroid that you might hear of hydra or one of these other cnidarians that grows on rocks, and then you sort of peel that little tube off the rock and let it drift around in the water column, and that's pretty much what a siphonophore is. The next phylum that I work on that also is relatively common and yet unfamiliar are the ctenophores, and cteno means comb, and phore again means bearing. So these are the comb bearers, and the combs are these little paddles that are arranged in 8 rows around the body of this guy. And in this case, the mouth is up here, and these tentacles are streaming along. And so they use the cilia to swim through the water column. They use them for a bunch of other things as well. So this guy just paused to put out his tentacles. These tentacles have sticky cells. They have gluey cells on them as opposed to stinging cells. And so when some kind of a prey item bumps into that, it basically gets stuck, and the ctenophore can pull that around and transfer it over to its mouth. And I should thank, this is a video that NHK took. The rest of the shots are stuff that we did, but this is from an NHK thing that came to film. So anyway, it's transferring that little thing into its mouth. So one thing to notice about that picture, the mouth was at the top, right, when it was hanging out, and then it swam around with its mouth first and transferred the thing. If you look in biology textbooks, invert zoo, I don't know if any textbook people are here, but the ctenophores always get this sort of backwards picture of them because people don't really know what to make of them. They literally don't know which way is up, which side to put up, and so they try to turn them into a jellyfish. So here's that same guy that we were just looking at the video, or that first shot, and so they turned it upside down, put the mouth down here, and make the tentacles go kind of up and drape down, because the only thing that they are familiar with is this jellyfish, that swims through the water with the mouth. But ctenophores actually swim through the water with their mouth first, and that's kind of how they encounter the environment. They're not really related to cnidarians and to these other jellies at all. It's more convergence that a lot of the organisms that live in that environment, they get transparent, they get fragile, they converge as opposed to sharing some kind of ancestry. So this is, again, one of the more familiar kinds, but actually in the deep sea we find a lot of different shapes and sizes and colors of them. This is a particularly fragile one that's from about 3,000 meters, and that one does seem to have a problem either with the temperature or some kind of pressure difference, because it just sort of melts once we get it onto the ship. So I had to take this picture just immediately upon getting on the ship, and then you'll start to see little wounds. You can see there's pretty much nothing to it, just these little tiny canals. This one is sort of fun. This is about 1.5 millimeters long, and we actually get this one scuba diving. This is a shallow water tropical one. But again, they have all different kinds of shapes and different sizes. Some of them don't have tentacles at all, and they're predatory on other jellyfish, so they just engulf them, and their mouth actually has little serrated cilia that act like little teeth. And here you can see, again, the iridescence in these pictures is just from the white light that we're shining on it being split. It's not bioluminescence. That's another thing that you'll see in textbooks, is like the rainbow bioluminescence of ctenophores or something like that. And that's all just from the white light in the photographs. So some of these different body forms have evolved, and it's hard to make sense of them from a line drawing, let alone or from a photo, let alone a line drawing. But here's an example of how this lobate form feeds. So this guy's flying straight out at you. This is sort of the last thing that a copepod might ever see. And so here are the lobes stretched out like a biplane, and they have these little sinuous called auricles that move up and down in between those lobes. And so when some kind of a prey item goes, normally it would just go straight through those lobes, but because of these auricles, it gets disturbed and it jumps up and it sticks on that lobe. And then, like we saw with the guy spinning around, this guy can transfer the prey over to the mouth and sort of down the hatch. So ctenophores, they're super fragile. There's no way to get them with the net, and so we need to use the ROV. And one thing that's interesting about these is that the new species that we're finding are not just, species B of some previously known group. When we look at the molecular tree and even at the morphology, we're getting completely new lineages of ctenophores. So these are new families in a lot of cases that we're having to put. And then we do get some where we're getting new species within a known lineage. But a lot of the restructuring of ctenophores needs to be done at the very basic level. So I have a lot of homework in that regard. So this idea that people have trouble understanding or appreciating ctenophores because even the textbooks are not really conveying such a good image of them because they just don't have firsthand experience with them. It's not just a problem for sort of school studies. It's actually also a scientific issue. This is a paper that was published in 2002, which is sort of my maybe least favorite paper ever, I don't know. But in this case, it's a fossil paper, and these fossils have been attributed to sea pens, so these sort of cnidarian things with polyps. Their mouths are up here and their foot is sticking in the soil. And this guy's hypothesizing that they're somehow linked to these fossils, which may or may not be related to ctenophores, purely because of this sort of, parallel morphology between the two types of organisms. So this paper got published. I hoped that it would die sort of a quiet death and nobody would notice it. But actually, in 2006, that paper was cited as a link between these fronds and the ctenophores. They're saying that they were probably benthic and used their ciliated rows for suspension feeding. So they're basically saying that ctenophores are using these comb rows to somehow filter particles out of the water because there's a link between them and these polyps or something. So, it's sort of bad science being perpetuated by other high profile, in science. It's not just some obscure journal. Anyway, so I entreat you to try to understand ctenophores on their own terms, not force them into a box, whether it be a jellyfish or a sea pen, that may be more familiar to you. Yeah?
Have they been sequenced?
Oh, yeah. So
wouldn't that be evidence?
Oh, well, the fossils aren't sequenced. The fossil,
No.
they can say whatever they want. Right, yeah, they've been sequenced, and I actually don't have a slide of it, but we did sort of a tree of life paper that tried to look at the relationships between ctenophores and jellies and sponges and bilaterians. And so those have a transcriptome actually sequenced, not just a gene or two, but thousands of genes from different ones. So yeah, we do have a lot of molecular sequences from them. All right, so those are some of the inhabitants of this environment. And now I'm going to talk about some of the adaptations in particular. And I'm personally focused on the sort of bio-optical properties of these animals. And there's some different ways that you can achieve color in the ocean. Pigmentation is what we used in our clothes and ink that we see on the paper are all achieved through pigmentation. And this is color that is through differential absorption, so absorption of different wavelengths. So you can't really use that so much in the ocean to get coloration, right? Because you have blue light to play with, and I'll show you an example in a second of why you can't do too much with it. But anyway, fluorescence requires you to provide some external light source on it. So with fluorescent light, if you shine a blue light on it, or like say your dark light on it, then you'll get a different color of fluorescence out, but it's only there as long as you're shining that light on it. Bioluminescence is this chemical reaction that's happening in the organism where it's actually emitting photons based on chemical energy. Okay, and sometimes these two are related, but they are distinct properties, and a lot of times people use those terms interchangeably, but they're not. So just briefly about pigmentation, we do see a lot of things that look to us like brightly colored organisms down in the deep sea. And so you're thinking, why would you be red? Why would you be bright yellow? And one thing to notice is a lot of them, the red is restricted to the stomach area. So this one actually is a really neat ctenophore that is also an undescribed species, but it has this sort of purpley color, and then in the middle it has the same red color. And there's even worms that have these orange guts. So I'm going to show you a little bit about why you might have that red. So here's essentially deep sea light simulator, which is available at Office Depot. And the light that's down there, again, it's filtered down to this uniformly blue light. And in here, you know, what do you see? You basically see the sort of dark circle. In the white light environment, that dark circle is actually this sort of bright red pigmentation. And the reason that it's red in white light is because it's absorbing all the blue light, and red is what's left over. So when you introduce that pigment into a blue light environment, it pretty much disappears. So red in the deep sea is essentially black. So if you pigment your gut red, you can protect yourself by, if you eat something that glows with bioluminescence, it's going to absorb all those blue photons and basically make you black. So I've talked a little bit about bioluminescence. Here's an example from this ctenophore. I touched it once, and these cascading waves of light come off. So this is sort of the nervous system transmission of a signal that in turn triggers light output from that ctenophore. And so in sort of generic terms, this light output is caused by the oxidation of this luciferin molecule. So the luciferin is the light emitter, but it sort of changes from group to group. And then the luciferase, or the photoprotein, is the thing that makes that oxidation happen. Okay? And so if you look actually at the distribution of the luciferin, so these are just the light emitters that we know from the deep sea, there's really only a few that are used by a large number of organisms. So each one of these groups has evolved its own luciferase or luciferase equivalent, but a lot of them are using the same exact light-emitting molecule. And we have some studies about why that might be. The other thing to notice is that bacteria, a lot of people feel like bioluminescence, they automatically assume it's caused by bacteria. There's actually really only a handful of things that use bacterial symbionts to produce their light. The rest of these guys are doing it on their own. So to sort of back up and look at the whole animal tree of life picture, and don't worry about reading this, but look at the blue on here are different groups where bioluminescence has evolved in the ocean. So you can just and this is from bacteria up to vertebrates, and you can just see it sort of popped up all over the place in that tree. So if we look in a little bit more detail, bacteria, amoeba-like radiolarians, dinoflagellates, the cnidarians that we're looking at, and then a bunch of worms can all be bioluminescent. Snails and mollusks like squid and even clams, and then all the crustaceans and copepods. Echinoderms, so sea cucumbers and things like that. No sea urchins that we know of are bioluminescent, but the rest of them have at least one member. And then sharks and fish and things like that. So there's no plants in here. There's no green plants. There's just some algae. There are no mammals in there. Those are some of the notable exceptions, but otherwise there's sort of bioluminescence has popped up and re-evolved at least 40 times probably independently across this tree. So why so popular? What is so great about bioluminescence? Well, one thing is that it's really energetically efficient, and so it's a way for a very small organism, like literally a 1 millimeter long organism, to communicate with something that may be meters away. You know, a giant fish pretty far away can see the light that's produced by a 1 millimeter organism. So it's a really efficient and effective way to communicate. If you tried to do that signal chemically or acoustically, the chemical signal would take too long to propagate and be too dilute, and the acoustic signal you would have to have some kind of hard structure or gas bladder to produce it. So I've sort of categorized, to try to sort of summarize a bunch of the functions that bioluminescence is serving, just to show you the diversity of things that organisms use it for. You can use it to startle. You can use it as a camouflage, which I'll show you some video of this in a second. But basically, a lot of things have eyes that are looking straight up. I don't know if you saw that that fish with the rotating eyes inside of its head. I was going to get some video of it, but I forgot. But anyway, a lot of things look up, and they just look for a little shadow passing overhead and that dim light that's coming down. And so animals counteract that by putting out their own little cloaking device, which I'll show you a picture of. A lot of things will shoot out a little particle stream of light while they swim the opposite direction. And sometimes they'll even leave off like a lizard's tail. They'll leave off a tentacle or something that glows while they swim away. And there's some other hypotheses for these sort of defensive purposes of bioluminescence. Things also use bioluminescence for offense, obviously, to anglerfish, the thought of attracting prey like moths coming to the light. And then there's some that have like a little red light they can use like a sniper scope, basically, where most organisms can't see that red light, but they can see it, so they shine it around and use it to have their own special ability. And then a few things use it for mating, but actually not as many as you might think, because the terrestrial example of fireflies is so familiar to people, but there aren't that many things in the ocean that operate in that same way. So just some examples of organisms using these different functions. One of them that uses that little cloud of bioluminescence. This is the vampire squid. And at the tips of the tentacles here are these sort of white patches, and those are actually bioluminescent particles. So again, here's some of the Japanese video that they filmed in our lab, so that it's turning its little umbrella inside out, and you can see at the tips of those tentacles these glowing things. And those particles actually will go out into the water to make a cloud of light. So a lot of organisms do that. This is a close-up of the tentacle, and so you can see where the suckers are, and then these particles around it. And actually, there's another cephalopod that has where the suckers themselves are actually glowing. And this was one that was immediately picked up by a lot of the creationist websites as an example of something that, it couldn't have this intermediate function because it makes the suckers useless. So a lot of these bioluminescence examples seem so unusual and so weird that they couldn't have happened gradually over time. But partly just by seeing how many times it's popped up independently across that tree and seeing all the different functions that it can serve, and then also seeing that the luciferins, or the light emitters, are sort of shared through the food chain. And so you don't have to reinvent bioluminescence from scratch. You can reinvent it by taking that luciferin from something that you ate and putting it back to your own purposes. Another example that we recently published about was these green bomber worms. And these guys are polychaetes that have these little fluorescent kind of globules attached to next to their head. And when you disturb them, these pop off into the water, and those things are bioluminescent. Polychaetes actually have a lot of different kinds of luminescence. This is a planktonic polychaete called Tomopteris. And this is one of the only things, it's probably the only one, that has yellow bioluminescence. And so here's the body of the worm down here that you can sort of barely see in the antennae, and then it's shooting out this cloud of these sort of golden particles. Most of the rest of bioluminescence in the ocean is blue-green, or one of those wavelengths. So this is a really unusual one, and one of my students is trying to figure out the chemistry of that worm. So another function, these are sort of these defensive startling or smoke screen kind of effects, but the other one is this counter illumination, which is a camouflage. And so this is a tank set up with a camera shining up, looking up from the bottom, and a dim light coming down from the top. And there's two fish in this tank swimming around, and these fish have these photophores on the bottom. So you can see when it cycles back around, we turn the light on, this fish turns on its photophores on the bottom and basically disappears against the background. And I sort of laid on the floor underneath this tank looking up. When the fish turned on its light, I could not see it from, a meter away. It just, perfect cloaking device. Then when we turn the lights off, he's like, uh-oh, and then realized that now he's exposed, and so he turns his light back down. So organisms will change the intensity of the light and the color of the light depending on the conditions to match that downwelling light. So the last example of a function is sort of this anglerfish. And one thing that we discovered was that there's another organism that uses an anglerfish type mechanism for getting its dinner. So do you guys remember what this is? All right. So it's a siphonophore because it has that tube. And it has these swimming bells here and all the stinging and feeding apparatus down there. This is one that we've only found 1,600 meters or greater, so it lives a mile down. But it also specializes on fish, and fish are not super abundant down there. there are some fairly small ones. And also, unlike the one that I described that's sort of 40 meters long, has this big web of tentacles sticking out, this guy is sort of a slacker and just is sitting there with all of its tentacles pulled up next to its body. So if you're living in this environment where there aren't many prey and you're not really making an effort to feed, how can this be a successful fish catcher? Well, the key is that it has some specialized structures next to its stinging cells. So there's a transparent stalk here and this bulb at the end that is filled with bioluminescent material. And this one actually happens to have this fluorescent coating around it as well. And so when it's sitting down there, it's flicking this little thing, which also glows. And so we're pretty sure that this is how it can draw fish over to take an interest in it. So I don't usually like wordy summarizing slides, but just as some sort of take-home lessons from this. One thing is that there's different constraints when you're operating in this environment, you know, this mid-water environment. There's sort of different constraints on the evolution than there are on a lot of marine species that are more familiar from the intertidal, like barnacles or things like that. You don't really have any pressure to go necessarily bilaterally symmetrical. You can be non-symmetrical or radially symmetrical. You have low food and the sort of low metabolic rate, so you can have kind of a long-term view of feeding. So you could feed, you know, once a year in some cases and be just fine. I think things in the deep sea also are fairly long-lived. We get very, very large siphonophores and ctenophores and jellies down there, and we don't actually know what their lifespans are, but it's got to be on the order of decades. So it's not this quick high turnover thing like you might find in some of the shallow jellyfish. Again, the water, when you get down there, it doesn't have any plankton or algae in it, so it's very clear. So a bioluminescent signal can be seen 50 or even 100 meters away. So light itself is a very efficient form of communication for both offensive and defensive purposes. And then the other thing that I like to point out is we think of the deep sea as, you know, this final frontier. It's as remote as outer space. But it's actually, again, one of the major habitats on the planet. And also you can find new species almost literally a stone's throw from San Francisco, from Manhattan Island. You can just go out from one of these major cities, and as soon as you get offshore and go down a little bit, there's this whole new realm of diversity that's just waiting to be discovered. And then again, we have an internship program at MBARI, so if you go, there's a summer internship program, mostly for sort of college, undergrad is the majority of the level. And then I have some more information about bioluminescence and some of these figures and more detail on my webpage. So I'd be happy to take questions if you guys have any. Thanks.